Class starts soon, everyone. Now look at these grasshoppers. Is exactly the vibes that I am wanting to convey here. And holy shit. Buddha 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 boy. Thank you so much for the resub. Absolutely fantastic. It is I. Ooh, ooh, ooh. Amy is here. It's her. Amy. We love Amy. Cancer, sex, cells, and germ. Cells are going to be the bulk of what I'm going to be talking about today, and it's going to be quite a ride. It's going to be fantastic. It's going to be crazylicious. I'm pretty sure probably all of our lives have been affected by cancer in some way. It's a very heavily researched yet incredibly mysterious set of diseases that can stem from a variety of things. But what all cancers have in common is the failure of cell cycle regulation. All cancers involve some sort of unregulated cell division where a population of cells is naturally selected for their survivability specifically. Um, and then the underlying mechanisms of this really, really horrifying disease can be genetic, epigenetic, environmental, but usually all of those things at once sort of play a role in the pathophysiology of cancer. It's a lot of things. It always is. I will be highlighting a specific subset of cancers called germ cell tumors, which arise from cells in the embryo that are supposed to eventually become the gametes or egg and sperm. Hi, Kurt. Don't worry, you didn't miss anything. You just missed me being scared that the wind is gonna knock the power out. And worried that my green screen is gonna, topple on top of me. But it's okay. We will persevere. Um, but yes, all cancers have those cell cycle regulation things in common, and then specifically talking about germ cell tumors, that arise from cells that are supposed to be egg and sperm. And as shown here in this beautiful picture, um, germ cell tumors they're not necessarily just in the gonads, just in ovaries or testes. They can also be um in the abdominal cavity. The mediastinum is the lining of your sort of chestal area. And they can be in your brain as well. So and since the reason they're still called germ cell tumors when they're there is because we name them based on the tissue of the cancer itself and not necessarily where it is. We don't really care about that as much as we care about the cells that make up the tumors. Um, I like to joke that I have a degree in cancer because the pathophysiology of all of these diseases, they're all really intimately connected to molecular cell and developmental biology. The M, the C, and the D. Um, so I'm gonna slide into this discussion by defining, what is cancer even referring to? What causes cancer? And a little bit of like who gets cancer and what are certain things that can predispose you to developing cancer. And of course, throughout the lecture, I'm just gonna sprinkle in some really, really fun MCD concepts. So we're gonna go pretty heavy into cell biology. We will be very activated while I talk about the cell cycle, which is the cell's mode of making copies of itself, growing and proliferating, of course. And then we're gonna see sort of the properties of cancer cells, what makes them so different from our regular cells that don't hate us so much. And then, of course, we're gonna sort of do a little bit of cancer cell classification, very, very tiny amount. And of course, we're gonna talk a lot about developmental biology, which, if I'm being honest, developmental biology is just cell biology plus molecular biology. It is a perfect mesh of the two of them. That's why it's so goddamn complicated. We're going to talk about how the germ cells are supposed to form, how egg and sperm are supposed to happen. We're going to talk about how these pre-germ cells migrate during development. And then we'll talk a little bit about the cell signaling that is involved in all that good stuff. Naturally, we cannot escape genetics, ever. We'll talk about the genetic changes that take place during the pathophysiology of cancer. Of course, this plays a huge role in those diseases. We'll talk a little bit about the epigenetic changes, which are changes that are not mutations in the DNA itself, but rather the things that interact with the DNA in order to turn genes on and off. That plays a huge role in cancer. And then maybe I'll go a little tiny bit into the specific genes that are involved in certain parts of cancer development. And naturally, our focus of today, germ cell tumors. We're going to talk about the types of germ cell tumors, how messed up they are, what can potentially cause them, why they happen, because why any of this, but also why germ cell tumors. They are like quite the aberration. And we're gonna, yeah, just the biggest why do these happen. These tumors are absolutely insane, and I'm gonna show you some of them. So basically the order is gonna be cancer, germ cell tumors, and then fucked up germ cell tumors. That is the itinerary. So cancer isn't just one single disease. There's over 100 different cancers just in humans, and they all have different symptoms, cells, presentations. So, I'm sorry to say, but a cure for cancer is impossible. That's not a real thing that will literally ever happen. We have to have multiple cures for the many different types of cancers that are out there in us humans. Um, so there are a lot of things that can increase your risk or your predisposition for cancer. And these are the main environmental risks to predispose you to developing cancer, developing really, really poor cellular regulation. Obviously, tobacco smoke is number one, surprising none of us. 90% of all lung cancers are caused by tobacco smoke, even today. And smoke has about 70 carcinogens. Yes. Is that right? Why do I feel like that's wrong? No, that's right. It has about 70 carcinogens. And the graph that I have inset in this other graph, this is showing the packs of cigarettes that specifically men, this is just men for some reason, um, consumption of cigarettes and then lung cancer deaths. And you can see maybe around the early 70s, late 80s, that's sort of the advent of anti-tobacco campaigns. This is in the US also. So it's anti-tobacco campaigns in the US. Less cigarettes are smoked, less lung cancer happens. Wouldn't you know? Also, the use of the word tobacco is intentional in case anyone is thinking, like that there's a difference between cigarettes and tobacco. Apparently, there is not. And rolling your own cigarettes or smoking hookah, whatever, is not going to lower your chances of developing cancer. Tobacco has inherent carcinogens in it that they're just present in the plant, and then it's also present in the smoke. So there's no form of tobacco that doesn't pose a significant risk to you or the people around you, just because of the inherent presence of the carcinogens. I did not know that. Obesity can increase your chances for developing cancer. On the outset, it's like if there's more of you, then there are more chances for cell divisions to go wrong. That's actually why taller people have an increased cancer risk versus shorter people, literally. Um, and so BMI, of course, is a measure of it's your mass over your height. So the more mass you have in your height, the higher your BMI is going to be. Hell yeah, short people privilege. We're immune to cancer. No. um, just for reference, I'm 5'6", 145 pounds. My BMI is 23. That's normal. 27, you're getting to the overweight section. 30, you're getting to the obese section. And apparently, each increase of your BMI by 5 raises your cancer risk of 10%. So, obesity also comes with a lot of other health problems, of course, like just like the systemic and chronic inflammation, which also creates an environment that cancer really likes. Um, so yeah, radiation is another duh kind of one because these radioactive materials really mess with the chemicals in your body. They mess with the DNA in your body, and it causes up to 10% of cancer cases. Um, eh, Radioactive materials do this thing where they break down our chromosomes and they really mess with our DNA. So, in a specific way, so that certain genes that are supposed to help repair DNA actually get turned off as a result of interacting with a radioactive substance. Messed up. And once that happens, that means that cells can just continually accumulate mutations. If you knock out the thing that was supposed to repair your DNA, that's what can happen, is this accumulation of mutations. Don't forget about that. It's a really important part of cancer. UV light and radon are just some examples of radioactive things. Infection can also cause cancer, about 18% of cancers. HPV is a pretty well-known viral cause of cervical cancer. And it may seem like it's not super surprising that viruses can cause cancer because they do go in and interact with our DNA, but bacteria and some parasites can also uh, mess with your genetic code and also increase inflammation, which again, cancer really likes inflammation. This is a really nice visual that just sort of represents how much of cancer cases are caused by environmental factors and how much of them are genetic. The right side here sort of breaks down like, give me my pen. Yes. The right side sort of breaks down basically what I just said. In my mind, I did group like diet with obesity, but it's listed here as like a separate thing. So having a really terrible diet, eating lots of sugar, lots of preservatives can predispose you to cancer. And then also drinking alcohol can predispose you to getting cancer. Sometimes the word environmental is replaced with the word preventable, uh, as in like there are lifestyle changes that you can make in order to lower your risk for developing cancer. Why alcohol? Uh, because it is I'm not gonna say it's exactly like eating a lot of sugar and like fueling cancer cells, but it's a little similar to that concept of like putting something in your body that predisposes your cells to not dividing correctly. But specifically, like if you think about it, your liver is doing a lot of work. So I think that um in the event that an alcoholic develops cancer, it's usually like hepatic cancer because, of course, those cells are being forced to do a lot of work and they're dividing more. And anytime you have way more cell division than is normal, that very much predisposes you to cancer. Um, so, consuming a lot of alcohol would be like a preventable, like considered a preventable cause of cancer. And that's not to downplay that addiction plays a role in a lot of these, I mean, alcohol and obesity. But it's just compared to the inherited and the sporadic mutations, these environmental, uh, when compared to that, environmental factors are preventable because you can't always control what you inherit. And then the left side here is just cancers that seem to have a pretty high heritability or genetic cause, testicular, thyroid, and laryngeal, your chances of developing those are greatly increased if you have a first-degree relative that went through having any of those cancers. So there are very, very specific and special hallmarks of cancer cells, tumors. They all have all 6 of these qualities. First and foremost, cancer cells will continue to grow and divide without signals that are telling them to do that, and despite signals that are telling them to fucking stop. They are dead set on survival. They do not listen to any signals that go against their extreme desire to survive. And I mean, in a normal cell, there's like a choose your own adventure kind of thing. So if there's DNA damage, for example, that's usually what causes the cell cycle to stop, is there's some sort of damage. So these factors will stop the cell cycle, and what can happen, choose your own adventure, we can repair the damage and then keep going and everything's fine. Maybe the damage is irreparable, so we put the cell in quiescence, or in some cases senescence, which is like that cell is never ever going to divide again, but it's just going to hang out and sort of do its thing until it's time to die, at least. Reporting in from the sickest you've been in years. Oh no, what do you have, like the flu or something? Is there something going around? That is terrible. Glad you're glad you're well enough to type out a sentence and come say hi, though, Dave. Hello. The last thing in the choose your own adventure is they'll just kill the cell. And that's a good thing. We want to kill cells that have irreparable DNA damage, because then it can't pass on its bullshit its bullshit broken DNA. So cancer cells literally ignore all of those choices, and they even become literally immune to apoptosis or immune to cell death. Even if so that means that if a cell has damage, usually cancer cells have DNA damage, they don't care. They're not going to die. They just keep replicating, and they keep passing on the damage, and as a result, also making more damage. Never heard senescence pronounced before. Oh, I love it when that happens. I love it when I hear a word out loud that I've only read. Here's hoping I pronounced it right. Senescence. Also welcome, friend. So yes, cancer cells ain't no cancer cell gonna undergo senescence. They're gonna survive. They only care about survival. To add fire to the fire, cancer cells are immortal. Just like we have, they become cancer stem cells, basically. And we do have a lot of stem cells throughout our body. We have them in different places. We have hematopoietic stem cells in our bone marrow that make all the blood cells. And we also have some in our intestines to make intestinal cells. Like, we already have our own populations of stem cells. But the thing is, they only make a specific subset of cells. Hematopoietic stem cells will only make the blood cells, nothing else. Oh no. The sore throat is my least favorite. This sounds so terrible. I hope you recover quickly and you don't have to deal with that for another week. That would just be, I would commit Sudoku. Don't go blind. Oh god, it gets worse. So we have stem cells, and we call that being multipotent, when you have a stem cell that can only produce a specific subset that's multipotent. And cancer stem cells are also multipotent. They can produce, a subset of cell types, but they're all, it's cancer, so they're all messed up. Angiogenesis. So a lot of times a tumor will vascularize itself. It will give itself a blood supply. That's what angiogenesis is. And that way they can better sustain themselves at your expense. It's so great. And lastly, cancer cells have the ability to invade other tissues without being detected by the immune system. And this is usually through the blood, the circulatory system, or through the lymphatic system, whichever highway they can get on to invade other tissues. We say that a cancer has metastasized when it has spread beyond its original tissue. And that's really bad. That is, when cancers metastasize, that usually, that's lowering your chances. That's making your prognosis worse when cancers metastasize. This figure here is showing us just the basic evolution of cancer. So on the left, purple, we have a cancer stem cell that is not only making its own progenitors. Progenitors are like the name of the cells that come right after the stem cell. It's making its own progenitors and it's self-renewing. It's making more of itself. And it's just continually cloning itself. And then at some point, both the cancer stem cells and the cancer cells, the progenitors, they gain the ability through a bunch of mutations probably. They gain the ability to enter our bloodstream or our lymphatic system, evade the immune system, and take up space in other organs. They metastasize. And again, these are in every single cancer has all 6 of these things. Even if a cancer hasn't metastasized itself, they all have the ability to do that. A lot of them do. I don't know specifically that almost all cancer symptoms start with unexplained bleeding, but a lot of them do. And yes, I think it could be because they are trying to make their own janky blood supply. Or it could be your body, like, because even though they're evading the immune system, your body kind of sometimes does still feel like, yo, something's up. So it could also be increased bleeding as a result of like your immune system is trying to react to something, but just doesn't know what the hell it is reacting to quite yet. Evolution. It's not generally something that I care about because I am M-bio, the superior bio, not E-bio, the poor man's biology. I'm just kidding, don't hurt me. But evolution. It is happening in our bodies right now, all the time as we speak. A population is the smallest unit that can evolve. So like a single individual, one person can't evolve. That's not how it works. But we have populations of cells inside of us. So those populations of cells absolutely can evolve, and they are doing it all the fucking time. Electric bio. If that were actually it would be so much cooler. But no, evolution. Boring, unless it's cells, unless we're talking about cells. It's ecology and evolution. That's e-bio. Yeah. Ecology is boring as fuck. Evolution is kind of cool. I do like it more when we're talking about the cells of our body, of course. But populations of cells can evolve through the process of natural selection. So here, we have a population of cells that I was just testing it. We have a population of cells that gains a mutation that increases its survivability. Whatever the heck that is. Maybe it's a mutation in a gene and the end result is that it helps it to evade apoptosis. It prevents it from dying. That trait, because it increases survivability, will be naturally selected. And as a result, cells will obtain more of those mutations that boost survivability. And while that's happening, that's also, there's going to be kind of this domino effect where it's going to continually sort of lead to mutations that, plot twist, increase survivability. Eventually, cells with that trait, with the increased survivability, they're going to outcompete the other cells. And this is, I mean, right here, this is kind of a very simple, not just like how cellular evolution works, but this is the pathogenesis of cancer that we're looking at right here. Um, and I do need it to be understood that the progression of cancer, it's so much more than just one gene being mutated. It's generally a lot of them. Uh, Genes, the magic fruit. Genetics, we have genetics and we have heredity. And all cancers, this whole circle, they all involve genetic changes, not just the hereditary factors. So they're all genetic, but not all of them are heritable. This 5 to 10% right here, where it says, the genes cause cancer, they're saying that these are hereditary predispositions to cancer. Another way of looking at this is that there's a percentage that's hereditary and then a percentage that is sporadic. So environmental factors that make you more susceptible to cancer, they're making you more susceptible to sporadic mutations that are happening in genes that are supposed to help regulate cellular growth and proliferation. Things that regulate the cell cycle. So yes, I just thought it was important. It's all genetic. It's all genes. It's all messed up genes. The cell cycle is how the cells pass on their genes. It's mitosis and meiosis, but we're looking at mitosis first. It's a really important part of the cell cycle, but it's a really tiny part of it. See right here. It's just this little slice of pie right here. The rest of this is interphase. And your cells spend way more time hanging out in interphase than they do actually dividing, than they do actually undergoing mitosis. And it's during interphase where cells are growing, they're preparing to divide, and they are replicating their DNA, replicating our whole entire genome so that its daughter cells contain the same amount of genes. So in G1, this is where we see a lot of growth. S stands for synthesis, DNA synthesis. This is where your entire genome gets replicated so that when the cell splits up, it has your normal original amount of genetic material in it. And then G2 is more growth and then actual like preparation for mitosis, preparation for actually dividing. Everything happening here. Everything happening here needs to be tight as heck. Everything needs to be perfect and everything needs to be correct all the time forever. There are checkpoints, in fact, that are happening at certain points during the cell cycle to sort of make sure that everything is all Gucci before and sometimes during cell division. For example, we can't go, there's a checkpoint around here, let's say. You can't go from G1 to S if you have DNA damage. That would be bad because then you are going to replicate the damage and then you're going to have two cells that they're both going to have that same aberration. We don't fucking want that. What we saw earlier with the choose your own adventure, this would be where that's going to happen, where it's like, do I die? Do I become senescent or however? Or are we going to fix the damage? It happens at the G1S checkpoint. Same thing with, I think G2 to M. This is another checkpoint where we're wanting to make sure like, all right, did everyone replicate correctly? Are we all connected? All the chromosomes like lined up or whatever? Like, is everyone cool before we divide? That's another place where, there are going to be checks for DNA damage. Even during mitosis, there's a checkpoint. I think it's during metaphase. Yes. Metaphase of mitosis, all the chromosomes are going to line up. There's a checkpoint where, if not everyone is lined up, if not everyone is making kissy being nice, there are proteins that will stop right there. They will arrest in metaphase because we need the chromosomes to they need to go to their own poles, and it needs to be even. Everything needs to be nice and even. So if it looks like that's not about to happen, this is another place where the cell cycle will just stop so that we can assess the damage, maybe fix it, maybe the cell commits Sudoku. Um and as you can imagine, oh my god, there's so many there's so many proteins, so many genes that are involved with the regulation of this. And we need them all to be able to do their job correctly, and we need them to be expressed in the right amounts at the right times. Everything just needs to be perfect pretty much. Um and there's one particular gene, one particular protein that's actually involved in every single one of these checkpoints. Uh and this protein is so important that we literally call it the protector of our genome. And it is mutated in about 60% of all cancer cases. P53. Protector 53. Just kidding. The P, I think, stands for probably something else. But this is the protector of our genome. There's a reason why such a large percentage of cancer cases, such a large number of tumors have mutations in P53. See, Pussy 53, you got it. I can't unsee that. It's gonna be Pussy 53 in my brain from now on, so thank you. So, Pussy 53, very important, if it's called the protector of our genome. Um, it I think this chart, what is it, 90%? Oh, yeah, small cell lung cancer, that's like 90% of tumors in that kinda cancer show mutations in P53. And as you can see, a lot of different cancers have mutations in this gene, which demonstrates really how important it is. So they all have some level of dysfunction in P53. Why? Well, it's vitally important, obviously, that our DNA remains undamaged. We don't want our DNA to be damaged. We want it to be nice and together. Um, and we also don't want that damage to be passed on to other cells. That is literally a one-way ticket to us just forgetting how to make ourselves. So, honestly, like, even the act of repairing broken DNA, those processes can also lead to mutations, even though they're trying to fix the problem. Um, and that, you know, can lead to accidental mutation. So it'd be nice if the DNA just didn't break in the first place, and if we just had a way to prevent any of this bad stuff from happening. So in walks P53 to literally do that. It acts at all of those cell cycle checkpoints that I talked about. It literally stops the cell cycle in response to DNA damage. And this is more or less this right here is more or less what can happen if DNA gets damaged. Some other proteins are recruited in response to the damage, and they all converge on the activation of P53. Don't worry too much about these other proteins. Just maybe this guy. Just consider MDM2 and how it inhibits P53. Interesting. But anyway, they all converge on the activation of P53, and then we have our choose your own adventure kind of thing, where P53 stops the cell cycle, assesses the damage, tries to repair it, and if it's irreparable, we go into senescence or we go into apoptosis. So that whole choose your own adventure, it's P53 that's pretty much controlling that. Oh my god. Dude, yeah, every time I go to a rave, I that MDM2, man. So P53. All of these actions on the right, what are they doing? They are preventing the development of tumors. That's why P53 is considered a tumor suppressor gene. TSGs regulate cell division and DNA replication in order to make sure that cells are getting the right, for example, the right number of chromosomes, make sure that they're getting the correct DNA, the right epigenetic modifications, because that's something that absolutely needs to be retained through multiple cell divisions, is the epigenetic modifications on the outside of the genome absolutely need to be the same in every cell. Because in a way, that's what identifies, for example, a liver cell from a bone cell is those epigenetic modifications. They have to also stay in place. So P53 and TSGs help make sure that that is going on too. Um, I have two other examples of tumor suppressor genes here. BRCA1 helps fix double-stranded breaks in DNA. That sounds really, really intense, and it is, but it's something that can happen when chromosomes are interacting with each other. Like they just kind of break each other a little bit. So BRCA1 is there to fix that damage. Um, y- you might recognize that as a gene that is associated with breast cancer because it is. It is mutated in a lot of breast cancers. It is literally called I forget what BRCA stands for. Breast cancer. It's probably something stupid. But it's named after breast cancer. We name genes after, diseases, but it is also, not just breast cancer, it can also be mutated in, what were the other ones? Ovarian cancer. Um, RB, the retinoblastoma protein. This was actually the first tumor suppressor gene to be, identified. It, as the name suggests, it causes retinoblastoma. And, what it does is it specifically stops the progression of, the cell cycle from G1 to S in response to damage. And it does that in a very interesting way. It will bind to, specific, like, promoters of certain genes, certain transcription factors, and those transcription factors are probably have to do with progressing the cell cycle. So RB literally goes in and goes, "Hold on, guys. We got a problem. We don't need you just yet. Let me do some damage control before you guys come in here and actually move us from G1 to S." So I think that's really, really cool. Um, there is another type of gene associated with cancer, and that is the oncogene. Um, they play a massive role in the development of cancer, but they're basically the complete opposite of tumor suppressor genes because uh in TSGs when you knock them out, um that is going to lead to a loss of cell cycle regulation, and an accumulation of DNA damage, and then eventually tumors. Oncogenes they're effectively the opposite. When these genes are unmutated, we call them proto-oncogenes, and they're totally normal, totally fine. They tend to promote cell growth, proliferation, and apoptosis. Maybe right out the gate, you could see why it would be bad if these were mutated because all of those three things, we want those to be pretty tightly regulated. We don't want anything messing with those processes. But when they're mutated, they become oncogenes, which are very yikes. They lead to unregulated cell growth, unregulated proliferation, and avoidance of apoptosis. The opposite of what we want. Um, and the so we call this with TSGs, we're seeing a loss of function, a loss of function in DNA damage mitigation. Whereas with oncogenes, again, it's the opposite. It's a gain of function, but it's a gain of a function that we don't fucking want. Uh, like we don't want increased growth and proliferation. We don't want any of that at all. That's a one-way ticket to cancer. Um, and mutations in those genes, instead of knocking them out, it makes them be expressed more. So we get increased protein product, we get increased mRNA stability, which leads to increased protein product. And then sometimes we even get gene duplication, which is literally it's exactly what it sounds like, where a gene gets duplicated. And when that happens, it just gets transcribed even more. It's great. It's not. We don't want that. It's bad. Um, I want to mention something called the two-hit hypothesis. which literally just means it's saying that two things need to happen for cancer to occur. Biallelic TSG mutation. Biallelic TSG mutation. So that would be you have one chromosome from one parent, one chromosome from the other parent. If both of those alleles for P53, for example, get knocked out or they get mutated, that is really bad. You don't have a functional copy at all. So that needs to happen, and you need the activation of some sort of oncogene in order to fully unregulate growth and proliferation. This isn't for every single cancer ever, but it is a hypothesis for the pathogenesis of like pretty much every cancer. Both of these things need to happen. It's called the two-hit hypothesis. An example of an oncogene is, as Kurt said, that drug we all took in college, MDM2. It is a proto-oncogene that normally, your Jinko genes. Oh my god. If I were in cancer research, you can bet your fucking ass I would name a new category of genes Jinko genes. So this proto-Jinko gene, MDM2, we saw it normally represses P53 activity, which we want. You know, it doesn't need to be P53 doesn't need to be active all the time. It only needs to be active when there's DNA damage. So we have this other protein, MDM2, to chill it out and make it, you know, go away basically until there's damage. So when this proto-oncogene MDM2 becomes an oncogene, when it mutates, it basically knocks out that tumor suppressor gene. Because if it's overreacting, then it's constantly targeting P53 for destruction, is constantly inhibiting it so that it can't regulate the cell cycle. And that is, that's like the two-hit hypothesis in one hit, because an oncogene is actively repressing a tumor suppressor gene. So yeah, oncogenes and tumor suppressor genes are the two main cancer-causing genes. There's one other, like, really, really crazy oncogenic change that can take place, and it's something called the Philadelphia chromosome. Why is it called that, you wonder? It was discovered in New Jersey. No, it was discovered in Philadelphia, and they just fucking named it that. But it's really interesting. It's really weird. So we've been talking a lot about cream cheese. That's where they first discovered it. Yes, was for the fucking cream cheese. You guys have the same brain. We've been talking a lot about mistakes that happen during like the cell cycle, and a major mistake that can happen during mitosis is something called chromosomal translocation. Um, that is exactly what is happening in the formation of the Philadelphia chromosome. A section of the Q arm, or the long arm of chromosome 9, and the Q arm of chromosome 22, they get swapped. And so then you have the Q arm of 22 is now down here, the Q arm of 9 is now up here. OG Philadelphia and Egypt are the US one that stole the name. The one in Egypt, yeah, they were doing, you know, chromosome staining and molecular research in OG Philadelphia in Egypt, yep. And they discovered the Philadelphia chromosome. So this is reciprocal translocation. They literally swapped. And that when that happens, that brings these two genes, BCR and ABL, literally right next to each other. And when that happens, this thing happens where you get leukemia. It's not funny, but you get leukemia because your body is like, oh, these genes are right next to each other now. What if I transcribe them all together to just make one fusion protein? It just does that. So the BCR-ABL fusion protein, it goes in and it messes with hematopoiesis. It messes with the creation of all of your blood cells. It usually affects the myeloid lineage here. So when you have this fusion protein, there tends to be like a large amount of those cells hanging around in your body. And not only will they be dividing like crazy, but the fusion protein doesn't care about DNA repair. So they're just gonna very quickly just constantly be passing on damaged and incorrect genetic material. Um, it's it's really great. So yeah, this is fucking wild. This is so wild to me that this is just a thing that happens. And then your body is like, what if this is weird. I'll just transcribe both of them together and we'll just have some fun with our leukemia. Like, it's wild. All right, now it's time to get married. It's time to marry our discussion about DNA and the cell cycle. Oh, earthquake. I didn't hear anything fall upstairs, so I think Onion's fine. That was a straight up earthquake. It was really easy for me to tell because I'm surrounded by a lot of very precarious instruments. Yeah, your body I was saying earlier that like they can evade the immune system, but your body can detect that there's some weird stuff going on. So it might do certain things to not directly attack it, but kind of be kind of a pussy and just figure out what's going on. The earthquake ruined our wedding. Our wedding of DNA and the cell cycle. And the result of that marriage is going to be so beautiful. It's going to be chromosomal nondisjunction, or chromosomal oopsie daisy, as I like to call it. Chromosomal nondisjunction, very important. This is when during cell division, a big whoops happens where one of the daughter cells takes an extra chromosome, and the other daughter cell is missing a chromosome. And so we saw earlier your entire genome, right before mitosis, it gets replicated. Your whole genome. So that means you got your 23 pairs of chromosomes. So 46 altogether because you get 23 from one parent and 23 from the other parent. All of that. Every single one of those. Your egg donor chromosomes and your sperm donor chromosomes, they all get duplicated. That's a lot of genetic material. I'm talking maternal 1 through 22, paternal 1 through 22, and the 2 sex chromosomes. They all get duplicated and they leave us with a total of 92 chromatids. Chromatids. You have sperm. If you have unprotected sex, sperm can live in your oviducts for quite a while. But okay, so I just really quick, I just want to explain chromatid. So I'm so sorry about this. I didn't do this. The discipline did this. For some reason, we say that a replicated chromosome has 2 chromatids. So the X that you always see when people are talking about chromosomes, that's 2 chromatids. And then we say that an unreplicated chromosome, like these guys, we say they're 1 chromatid. There's a reason for this, but it's not good enough that I feel like explaining it. I just needed you to know this information, and we're just gonna look at this chart. Number of chromosomes, number of chromatids throughout all of mitosis. So a prophase, we have 46 chromosomes and 92 chromatids, okay? In metaphase, they all line up. So pretend that this is 46 chromosomes and 92 chromatids and not 2 chromosomes and 4 even this fucks me up every time. And not 2 chromosomes and 4 chromatids. I don't know why they do that. They just do. Also, these are the same chromosome, okay? One is maternal, one is paternal, but let's say they're both chromosome 13. But because you get one from one from here, they duplicate. This is the same chromosome. It's a diploid cell. We are diploid beings, by the way. But anyway, I'm sorry. I'm really mad about chromosome and chromatid. I will never not be mad about that. You were just a sperm. Well, yeah, that's quite an existential conversation. Were you ever the sperm and were you ever the egg? We could say that you were both, or you could say that you were never either. You were always the combination of the two. You were never either of them. So existential. I love it. But anyway, I'm sorry. We're counting. We're doing fucking math. So they're all lined up. And then anaphase happens. And they all go their separate ways. Now we have 92 chromosomes and 92 chromatids. They're the same thing now. One chromatid per chromosome. They get separated. And now we have two daughter cells that are both diploid. They have 2N. N means the number of different chromosomes. So we have 23 different chromosomes. So we always have 2 of each in our cells. This is normal. And at the end of mitosis, we don't call them chromatids anymore, really. Even though they do here. They're chromosomes. Oh my god. You know how I heard this explained as like a house? A house can have one, let's say a house has 1 story or 2 stories. They're both houses, but they're into stories. So the house is the chromosome and the stories are the chromatids. I think that that's the best way that I can explain it. It just makes me so mad. So this is normal. Our diploid cells. This is mitosis. So these are our body cells. All of our somatic cells are doing this. My skin cells all have 46 chromosomes, 23 from my mom, 23 from my dad. Simple. It's fine. This is good. This is in contrast to our gametes, which we'll talk about later, but gametes are haploid. They have half the number of chromosomes. They have 23. Sperm 23, egg 23, 46. This is fertilization. So let's make our whoops. Let's make a whoops happen now. We're chilling in metaphase. Our chromosomes are lined up. And it's time for anaphase. We're going to split our chromosomes. They're going to go to their separate corners. And oops, these ones didn't separate. The maternal chromatids did not separate. This is nondisjunction right here. This is the definition of nondisjunction. How codependent of these chromatids to just stay together. This means that one of the daughter cells is going to have an extra copy of that maternal chromosome, and only one regular amount of the paternal. The other cell is going to have no maternal chromosome. It's going to be missing a chromosome. You're just thinking that's so codependent, right? It's like they just stay together. Like it's literally life or death. You're supposed to go separate ways. So these cells are aneuploid, meaning they have the wrong number of chromosomes. That's what aneuploidy is. This is yikes. This is pretty yikes. These non-disjunction mistakes in mitosis. But at least it's mitosis, you know? Like, it's our somatic cells. Oh, what's up, popcorn fart? Is indeed fucked. Mitotic non-disjunction, nothing to joke about. But we will anyway. But that is the beauty of mitotic non-disjunction. We have fail-safes upon fail-safes to make sure that, you know, we can either prevent this from happening in the first place, or maybe fixing it. Maybe, like, you know, apoptosis or whatever. Maybe we can get some of that going. That's what we hope will happen to the aneuploid cells, that they apoptose or undergo senescence. because we don't want these defects to be passed on. Interestingly, whether or not aneuploidy can lead to cancer is heavily debated in the field. But there are two things that we know for sure. The first is that nondisjunctions in somatic cells can certainly lead to cancer if there is an existing mutation. It acts as that second hit in the two-hit hypothesis. So, for example, we'll pretend this is the RB gene. On the maternal chromosome, it's mutated. On the paternal chromosome, it is good. So, you know, not the best thing ever, but we have a good copy. You know, we have a good copy that can make the gene and do what it needs to do. That is until nondisjunction happens. Because when the chromosomes line up and they duplicate, so the mutated chromosome is gonna, I didn't draw it here for some reason, but the mutated chromosome is gonna, this is a chromosome. It's gonna duplicate just like the good copy. So we have the bad and the good both duplicated, and they're supposed to separate. And then the idea is if nondisjunction doesn't happen, you just get this exact thing right here. You get, this is heterozygosity, basically, the two different alleles. But if there's nondisjunction, let's say that the good copy undergoes the nondisjunction, the two chromatids go to one cell, and that leaves the other cell with 0 good copies. That's bad. Now, it may seem like whatever, this one has like 2 good copies. Who gives a shit? There are a lot of shits to give because having the wrong number of chromosomes is just not good. Even if it's because you don't just have 2 copies of that good gene, you have 2 copies of every single gene on that chromosome. You don't know what's on that chromosome. There's tens of, I think chromosome 1 might have hundreds of thousands of genes. There could be a lot. You're not just duplicating a good thing necessarily. So it's pretty much never a good thing to have the wrong number of chromosomes in a somatic cell. And that's not to mention this other cell only has the bad copy. And what's going to happen when this cell does further divisions? The bad copy is going to get replicated. They're going to, and then all those cells are going to only, every single cell that is the daughter cell of this, all the ones down the line, they're only going to have the bad copy. Not so great, is it? That's bad. That's two-hit hypothesis, baby. This is the we're talking about the RB gene. This is one of the mechanisms by which retinoblastoma occurs. Mutation, and then loss of the good copy. This here just demonstrates pretty much exactly what I talked about, where we have on the left here, we have the 2 wild type copies. One gets mutated. And then what can happen is we have loss and duplication and loss of heterozygosity. That's what I just talked about, was loss of heterozygosity, making it so that there is not a mutated and a good, but rather only a mutated. You can also have mitotic recombination because, of course, inevitably, because these chromosomes are lining up during cell division, they're going to interact with each other. So you might have it so that the bad gene is interacting with the chromosome that has the good gene, and then it's like, here, you actually have the bad gene now. You have monosomy, which is basically the beginning of the process for us here. It's starting with a monosomy. That means you have 1 copy of a chromosome that you should have 2 of. You could accidentally straight up get the gene deleted on one of the chromosomes. Just gone. And then lastly, you can just have, I don't know, I think this is just saying that, like, you can also, have a mutation that ends up arising in the good copy. So we know, based on this, that aneuploidy absolutely plays a role in cancer, at least when there is already an existing mutation. The other thing we know is that, in regards to aneuploidy, is that cancer and tumors tend to be pretty messed up in their chromosome number. This makes sense because they don't have the machinery that is supposed to like, the machinery that we've been talking about that's supposed to repair damaged DNA and regulate the cell cycle. Those proteins that help with that are getting knocked out left and right. Um, so these 2 karyotypes, these were taken from retinoblastoma samples specifically. Um, on the one on the left, also these are metaphase chromosomes, that's why they're X's. This right here, I think what happened is, this chromosome has an extra P arm. So P is the short arm, Q is the long arm. It just got another one just put on top of it. That's weird. That's fucking weird. Um, it's like not even a translocation almost. It's just like it just made an extra one. It's absolutely bonkers. Um, there's also where is it? Yes, trisomy 6. There should obviously this should not be here. Chromosome 13 here. This is a messed up guy. It's not, apparently it's not a translocation, it's just a bunch of duplicated genes that just kept duplicating. So, different from chromosome 1 here because it was literally just a P arm of chromosome 1, boom. They're like, that's just the P arm. This is just duplicated genes that are extending above the chromosome. Bunny rabbit. Oh my god. Yeah, oh my god. Little Bugs Bunnies. Um, so, yeah, so this person, this, individual, this is not what their regular somatic karyotype looks like. This is the cancer cell that is in them because they are probably I'm gonna guess that they do not have Turner syndrome, that they are a regular XY male, but the Y chromosome was also just not passed on in these tumor cells, which make Y chromosome very small. Very small, not very strong. So that is how messed up these tumor cells are. They're so unregulated. They don't give a shit about repairing their DNA. On the right, we have an extra chromosome 1 here. And then this fragment right here apparently came from chromosome 2. Just a translocated fragment that plopped itself on the top of one of the chromosome 6s. And then we have only one copy of 16 and 17. We have a monosomy of chromosomes 16 and 17. As you can imagine, the initial non-disjunction that may have taken place that led to the creation of these weird karyotypes in these cells has sort of sealed the cell's fate in a way. Sealed their fate is just a complete failure in cell division and DNA repair. So they just keep getting wronger and wronger and wronger. That's tumors. They just keep getting wronger. Teratomas are some of the nastiest tumors out there, and they also show whack-ass karyotypes and messed up chromosome numbers, whether it's extra copies, missing copies, uh, of multiple autosomes or sex chromosomes. Like, they're just messed up, and those monsters, teratomas. Teeth! Exactly, exactly. So gross. Only freaks grow teratomas. Exactly, I was thinking the same thing. Only freaks and losers grow teratomas. How could you be so wrong in your cell division and also your development? Um, Speaking of development, those monsters, teratomas, are a subset of potentially cancerous aberrations that I shall go into now. So we're having a sex talk also. We're gonna have a sex talk right now. Because germ cell tumors arise from what are called primordial germ cells, and they eventually become the gametes, sperm and egg. They're also called sex cells. So, in the beginning, I lied to all of you with sex cells and germ cells. They're the same. I'm sorry. But anyway, that's what's pictured here. The primordial germ cells here, they are arising from, this is a very early embryo. This is probably week 2 or 3. The primordial germ cells are coming from specific cells in the zygote or at this point, maybe the blastocyst, called the epiblast. So those cells, they're like, I'm me now, I'm a PGC. They migrate, just like here, they migrate from the yolk sac to where the genitals will eventually be, and they become sperm or egg, depending on uh, protein expression, genetics, all that good stuff. Um, so germ cell tumors uh, are originating from those cells, and what makes them slightly different from the cancers that we've been talking about is the fact that they arise more from inborn failures during development. Uh, there's certainly genetic and environmental factors that are in play, um, but that is a defining feature. The fact that it is a developmental mistake, as you can see here. Just a little sneak peek. They're supposed to go here. What are they doing? Why are they going here, and why are they going here? Who knows? So think back to when you were a zygote. Think back to that period in your life. You're just a ball of cells, with nearly unlimited potential. If we're talking day 3, totipotent for sure. Any cell you want. Anytime after that, pluripotent. You can become a lot of different cells, basically. Any cell, at least in the human body. Eventually, those cells are going to need to figure out who they are. Uh, and at around 4 weeks, some of those cells from the epiblast right here, some of these cells are going to realize that they are primordial germ cells. They're gonna realize that they're eventually gonna become sperm and egg, and they need to migrate from where they are currently to which is near the yolk sac. They need to go from the yolk sac to the primitive gonad, or maybe where it's going to be. So they march on over. So this is this slice that we're looking at here. Imagine it's like like this way. That's kind of how we're looking at it, where this is the front of the embryo and this is the back of the embryo. So how the migration might look a little bit from this perspective is the cells are going to be moving to the back, to the hindgut, because they're kind of here. They're kind of in this area. That's where they start, and then they're going to migrate toward the hindgut, through the hindgut, and then to the where you would find eventually the gonads. Gonads, of course, being ovaries or testes. While they're doing that, they're listening to different signals that are telling them where to go, basically. That's called coming out of the closet. True. The hindgut is the primitive, so when you are developing, you're kind of just a tube. It's the part of the gut that's closest to the tail, that's closest to the bottom of the body. So that's kind of where it's going toward. It's going toward um around the tail area, maybe up a little bit, and then over to here. That's sort of what I mean by the hindgut. Um, so they're listening to different signals to make sure that they stay on the right path, to make sure that they go here and that they stop there and that they don't go anywhere else. That's one thing. And they're also listening for certain signals in regard to like what they're supposed to become. Because right here, they are pluripotent. And what that means is they can become any cell in the human body that is not extra embryonic. Totipotent means literally any cell ever, including placenta, trophoblast, the yolk sac, that kind of stuff. But they're pluripotent. They can become a lot of different things. So they're trying to listen to signals on what they're going to become to make sure that they become the right thing. And it's really important. These signals are really important. Every cell needs to go to the right place. They need to be in the right place. There's no room for messing around. And yeah, so another thing about pluripotency, if you're a diehard fan, you might remember this chart from my epigenetics lecture. But this so the pluripotent PGCs are demethylated. Pretty much their whole genome is demethylated. And what that means is it's very open at this point. It can express a bunch of different genes. Um, and it's a form of epigenetic reprogramming. And you can see here, CpG methylation, the higher that is, uh, the more closed the genes are, the more methylated it is, the more differentiated the cells are, meaning that they're becoming what they're supposed to be. So we see a higher methylation state in the somatic tissues right here because they're becoming, you know, the mesoderm, the ectoderm. They're becoming what's going to be skin and nerve tissue. They're committing basically. Methylation, you can think of methylation as turning genes off so that cells are being committed to what they're supposed to be. Because not every single cell in your body is going to express every single gene. That would make no sense. That would be like if bone cells secreted gastric acid. Like that makes no sense. PGCs have low methylation state. They're just kind of open. They can express a lot of different things. And it's not until they get toward birth for sperm people and toward puberty for ovary people that the methylation increases and you get more of a not necessarily committed because they are committed at this point, but more that they are what they are. So keep that in mind. The fact that PGCs have a low methylation state, that they're pretty free and pluripotent. That's gonna come back. um It's really when they make it to the gonadal ridge, so that's, what we're calling this area right here. Um, they're gonna receive signals that are sort of gonna remove their pluripotency, and they'll actually commit to becoming gametes. Um, that's closer to 7 weeks. So this is week 6, 7 is when the PGCs are at the gonadal ridge and they're starting to be committed to whatever they're gonna be, egg or sperm. Um, yeah. So if ovaries are happening, uh, those primordial germ cells are gonna start undergoing mitosis and differentiating into oogonia, my favorite word. And then that reserve of cells, oogonia, you can think of as like a reserve of eggs, kind of. Um, uh, they will eventually differentiate into a number of oocytes, which is the eggs themselves. So oogonia undergo mitosis to become oocytes. And that mostly happens before birth. And those gametes, these ones, so the post-mitosis, they're going to stay frozen in meiosis 1. That's like, they haven't even started it. They're in prophase 1 of meiosis, sorry. They have a lot of genetic material. They have not become haploid yet. And it is only during puberty, during menstruation, leading up to ovulation, that these eggs are actually going to complete meiosis 1, and then they're going to be arrested again in metaphase 2 of meiosis. Only fertilization itself causes an egg to complete meiosis. Only fertilization causes an egg to complete meiosis. Sperm are completely different, obviously. The spermatogonia, they don't really develop and start doing their mitosis thing until 2 months after birth. And then full-on sperm differentiation, so full-on meiosis, is not going to happen until puberty. So these are two very they're all germ cells, but they undergo very different processes. It's very interesting. And it's great. It's so simple. Just make an egg and sperm. Easy peasy. But we know that in reality, there are millions upon millions of signals, millions upon millions of proteins that are working to make sure that this whole process gets done correctly. And sometimes that just does not happen. So, germ cell tumors, they are classified based on their histology, based on how they look cellularly. And they're also categorized based on their prognosis. There are two main categories, the first being germinomatous. And the germinomatous germ cell tumors have a better prognosis than the other counterpart, and they have an undifferentiated histology. So their cells show more pluripotency markers. They are not like it's clear that they're of germ cell origin. We know that for sure, but they're undifferentiated. They're perpetually early in their development process. They kind of stay there, um, and uh, they're found. They're pretty much the same. It's just they're just named on where they are. So germinomas are found in the brain and mediastinum, which again is like the lining of the chest cavity. Dysgerminomas are going to be found in the ovaries, and seminomas are going to be found in the testes. Um, so these all I just showed the histology. I'm not a histologist. I can't 100% describe what we're looking at, but they are all very similar. They're the purple things, those are the nucleoli and the nucleuses that are stained. They're kind of weird looking. They are not supposed to I don't think they're supposed to be that big. Um and then the surrounding tissue, it's like semi-organized, but ultimately, like I said, undifferentiated. Um The other category is non-germinomatous, and these have a poorer prognosis, and they have a more differentiated histology. So we have yolk sac tumor, embryonal carcinomas, and the choriocarcinoma, as well as the teratomas. These top three are not, they're very, very, very rare. They're not as well studied as the teratomas. And they can all be found pretty much where germ cell tumors can be found, except the choriocarcinoma, of course, that tends to show up in the placenta. Um, but yeah, they're just not well understood. So I'm mainly going to be talking about teratomas and then germinomatous germ cell tumors, like in general. But teratomas, I'll just show you. Okay. Oh, can I do this? Oh, this. I hope you guys can see this blue lining right here. This is a sebaceous gland. We're in the ovary, okay? And this is a sebaceous gland. The red is follicles, oocytes, and then I believe up here, the yellow is epidermal tissue. So there's grossness. Exactly. Sebaceous means oil. Yeah. Natural skin oil is secreted by the sebaceous gland. So yeah, teratomas and other GCTs are yolk sac. These they're just rare, and yeah, they look like one of your work slides. Ooh, interesting. I wonder if do you know if it's a germ cell tumor or if it's maybe just tissue from an area? Amy's a hysterectologist. Oh my god. All right, so let's get to the root a little bit. Because the root cell of germ cell tumors has to do with incorrect primordial germ cell migration and differentiation. And that's how these tumors can develop in our frickin' brains. It looks more muscular. That's cool. I like your slides. Um, so something went wrong during their development that caused them to basically continue along the nerve that they were traveling. They did not get off at the stop that they were supposed to get off at. Um, and they just kept going. So, and this isn't an embryo, right? I mean, it's tiny. So the road from the gonadal ridge to the mediastinum or the brain is not it's not that long, like it's right there. Um so and they're not only going to the wrong place, but there's something that is preventing them uh from dying because these cells when they get anytime they leave the area that they're supposed to go, they're supposed to be killed on site. So there's something like in cancer that is making them avoid apoptosis when it's supposed to happen. All GCTs demonstrate these things here. They demonstrate some sort of cell cycle or apoptosis dysregulation. They show DNA damage. There is messed up cell communication involved. There is aberrant pluripotency, as well as epigenetic markers that are affecting the pathophysiology. And then, of course, sex determination pathways can affect GCTs as well. And with all of these, even though we're looking at incorrect migration, there's still genetics, all right? Like still, there's there's pretty much always going to be some sort of a genetic component. Because genes code for proteins that are supposed to do everything here. Especially help the cells communicate with each other, and communicate with their environment. Um, Genes also tell a cell when it is and is not time to shed their pluripotency. Remember that. And these, like if we're talking about germinomas here, they don't show a lot of differentiation, right? They, compared to the non-seminomatous tumors, compared to like the teratomas, they show an aberrant, expression of a gene called SOX17, for example. Now, with the aberrant SOX17 expression, these cells are needlessly and latently pluripotent when they are not supposed to be. And they resist differentiating. And so that's why, again, the germinomas with the lack of differentiation is because of these pluripotency factors. On a more epigenetic level, PRDM14, this guy helps to keep the cells hypomethylated or lowly methylated, which is a way of preventing them from differentiating. Because again, we see higher rates of methylation or genes turning off when we have differentiated somatic tissues because they know who they are. They know who they're supposed to be. But if you have PRDM1 that is keeping them hypomethylated, then they're going to keep not knowing what they're supposed to be. And it keeps pro-pluripotency pro-pluripotency genes, they stay expressed. There's also a connection between issues with genes that regulate sex determination pathways and GCTs. So people with disorders of sexual development, whatever that looks like, they're going to have a higher likelihood of developing germ cell tumors. Let's look at this timeline once again and insert the development of GCTs. So we're looking mainly at type 2 right here. We're mainly looking at like this group right here. Notice that there's this thing called GCNIS. Germ cell neoplasm in situ. That's the name they chose for it. In situ means where it is. It's a way to distinguish when cancers metastasize. In situ is referring to where the original tissue that it arised in, as opposed to the other places that it metastasized to. So it's saying that this is where this neoplasm started, was in the germ cell. And so this GCNIS is referring to a neoplasm or a tumor that is sort of a precursor to these guys. So this means seminoma, but it's referring to germinomas and dysgerminomas as well. I don't know if there's an opposite of in situ, but people say in vivo to generally mean like within life. Um, there might be a term for the opposite of in situ, but I think that people just speak in terms of like metastasis, like it's metastasized. And then we only care about the in situ. Possibly. I am unsure. Um, so this is a precursor again to all of these. It's a precursor to seminoma, germinoma, dysgerminoma, and it's a precursor to these rare guys that I said, and therefore to teratomas. Anyone who develops these starts with GCNIS. um, we can call it precancerous, basically, and it's developing. So right here, it's developing in weeks 4 through 8 of embryonic development. So not only do they all start with GCNIS, but they all start before birth. These things start growing before birth. And this is coinciding with the PGCs migrating toward the genital ridge, of course. So essentially when these conditions are met with inappropriate pluripotency, unregulated cell division, and incorrect migration, like the whole thing, then groups of these cells can develop into GCNIS as a precursor to GCT, germ cell tumor. GCNIS has nasty cells. They have double genome. They are polyploid, which, I mean, that's another way of saying double genome. They have multiple copies of chromosomes. And they retain the hypomethylation that we see in normal primordial germ cells before they're supposed to be differentiating. And as a result of all of these things, the chromosome stability is incredibly low. So there tends to be a lot of like gains and losses of genetic material. There is one chromosomal aberration that seems to be like responsible for the switch from GCNIS to germ cell tumor. And that is the acquisition of something called isochromosome 12p. This is it's eventually something that happens within a GCNIS and is subsequently seen in almost all cases of germ cell tumors. In the cell, we'll have this wimpy version of chromosome 12 that will just be 2 P arms on top of each other. Like, it's supposed to look like this with the P and the Q, but it'll just be 2 Ps, 2 PPs. And I believe it's an extra chromosome. So you have, in one cell, you're going to have the 2 regular copies of chromosome 12 and then this isochromosome, which is freaking weird. It's not well understood, but again, this is seen as something that is like it's like a transition point between GCNIS and full-on GCT. And there are some genes in this area that could contribute to the pathogenesis, like NANOG and KRAS. Those are both proto-oncogenes that are also promoters of pluripotency. Like I said, pretty much all GCTs have this. In the very few cases that don't have isochromosome 12p, they still demonstrate amplifications of genes located on the P arm of chromosome 12. So it's like they might as well have the isochromosome, basically. So after all that talk about and also GCNIS itself, the cells of the GCNIS, they don't have this aberration. It's only the GCT. So that's why they think there's like a transition thing that happens where the acquisition of this isochromosome is what leads to the development of GCT from GCNIS. But after all that talk about latent pluripotency, aberrant migration, and really, really messed up cell division, I think the next subject will make a lot of sense. and I might we might even get into some gross pictures. We might do that. The prize of today's lecture, teratomas. They are non-germinamatous GCTs that have a lot of different cell types, so they are pretty differentiated. They don't tend to have a lot of undifferentiated like the germinamatous ones. They are, boy, do they have developed cells. Teratoma means monster tumor or monster swelling, and they're usually benign but categorically disgusting because depending on the type, they can possess all of the germ layers present in a human embryo. And all our the germ layers, all our starts as endoderm, mesoderm, or ectoderm. And due to this pattern of pluripotency, undifferentiation, dedifferentiation, teratomas can have some pretty odd qualities. Surprise developmental concepts, though. More germs. Let me tell you about the formation of the germ layers, which eventually become our whole entire body. 17 days into our development, a groove called the primitive streak starts to form. And this is what establishes our body axes, our bilateral left and right, and where our head and our butt are. Because humans are deuterostomes, we start asshole first. So it's the primitive streak is going to start at the asshole, basically, and move up toward the head. We are, we truly, we are but a disc at this point. So let's flip ourselves over and cut ourselves in half and look inside. Again, this is like day 17 post-fertilization. A couple days after the primitive streak shows up, the embryo is going to undergo gastrulation, which is the formation of the 3 different germ layers. And that's a human being who deserves rights. I know, it's so cute. How could anyone wanna, you know? Uh, this 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 is a massive cells here, and the on the top here, we have the ectoderm. Um, ecto means outside, so it's gonna form the outside type tissues like skin and hair and nails and sweat glands and our enamel and most of our CNS. Then we have the mesoderm. That's gonna form our middleist stuff, like our muscle, bone and cartilage, connective tissue, fat, circulatory and lymphatic system, urogenital system, and the mesothelium, which is like the lining of our middle parts, basically. And lastly, we have the endoderm, which is our inside ear parts, like our GI tract, our bladder, all the epithelial linings that are within our body. That it also forms our lungs and thyroid, and then like the linings of those things as well. So as you can imagine, there's a variety of things that control this process. There's a lot of different signals going on, telling cells where to go, attracting them to certain areas, detracting them from certain areas. It all needs to be correct. I've been saying that the whole time. These things need to be done correctly. And when things go wrong, both germ cell and germ layer development, when things go wrong in germ cell migration and germ layer development, that's when we can get a teratoma pathology. So I'll start with the most common tumor that is found in newborns, which is the sacrococcygeal teratoma. It's a like I said before, this starts growing during development. So this probably starts growing as the primitive streak is forming. It's usually in the tailbone area, like when babies are born, they have a mass on their tailbone. It can also be internal, which causes more problems, but as you can see, these are the migrating PGCs. They touch the tailbone. They touch the tailbone like area. They go right by there. So it makes sense that in addition to the PGCs migrating there and getting trapped, you also have this disorder of like germ layer development in that area. Um and it has the tumor itself does have all 3 germ layers. Oh yeah. So it's not just the germ cell migration, it's those pluripotency switches that are not quite being correct. It's not fully understood why they form, but we do know that they are mature cells, they're differentiated, um, and they can sometimes have immature cells, which anytime there's immature cells, that tends to increase the chance of, like, malignancies. Um, there's also no formation of GCNIS, uh, so there's no isochromosome 12p, which further demonstrates that this starts growing, um, and developing before the GCTs that start GCNS. It starts before that. Um, because like this is saying, yeah, GCTs are 4th week, and what did I say over here? 17th day is when we have the germ layers. So it starts growing um before GCNIS would occur, which is very interesting. Um, the prognosis is usually good. Especially if the tumor's on the outside, it can be removed along with the tailbone. Usually the tailbone has to go with it. But more problems do occur if it happens to be like inside. It can mess with the urogenital um area. Um, there are also risks with the pregnancy of like swelling and edema, which can require intervention, but the outcome is usually positive. All right, this next picture is gross. This is a warning. At the end of this sentence, there will be a gross picture on the screen. This is the fetiform teratoma, which is exactly what it sounds like. It is a teratoma that has like embryo-like qualities and contains straight up like pretty developed organs or organ systems. Um, this one has straight up um ovaries. It has 2 adrenals. It has renal tissue as well. There's bone marrow, stomach. You see the intestines right here. I know, right? Like, I think this is the ovaries. I think these are the adrenals. Actually, those look more like kidneys. This is obviously the intestines. And bone marrow? Like, holy shit. Um, so this is mostly ovaries on ovaries. Yeah, yeah. Oh, I wish I remembered where this was taken from. Probably an ovary, though. Ovaries are really hot ticket areas for teratomas, of course. But yeah, really gross. This is mostly mesodermal, uh, with a little bit of endodermal spattered in there. And in the picture I drew right here, I tried to show that the endodermal cells and the mesodermal cells are kind of like uh migrating from the same place. They both come from the epiblast, which of course is also where PGCs come from. Um, so to me, this makes sense that like full-on organs from those germ layers can form together incorrectly in fetiform teratoma. Um, what if you had hyperthyroidism, but your thyroid was completely healthy and it turned out your ovary thought it was a thyroid and it was producing excess thyroid hormone? Wouldn't that be wild? It is, and it can happen. Uh, thyroid tissue comes from endoderm, which again, arises from the same cellular location as germ cells. Um, I'm wondering if, like, something causes these cells to accidentally migrate together, um, in this case, and I mean, I find this extra interesting because the thyroid, it forms in a completely different area, right? But it's along the same axis. It's in line with where the PGCs would go. So I don't really know why it's getting signals to form in the gonads. Like that is so weird. I don't get it. A nurse said another nurse didn't know she had a teratoma until she realized there was a hair coming out. It's okay. Wow, it was going straight through. Oh man, that is so nasty, dude. That is so gross. Oh, why our bodies do this? So nasty. All right. So the last three teratomas, those are mature teratomas. They have established tissue, adult tissue. It may be of, embryonic origin, but they are established differentiated tissue. Um, and they don't tend to be malignant, unlike in the immature teratoma, which are malignant. They make up about 1% of all teratomas. And the the thing that makes them different tissue-wise is that they they have immature tissue. So that's neuroepithelial tissue. That is not adult. That is straight up embryonic tissue. That shit is supposed to be completely replaced with different differentiated adult tissue at a certain point. But because it's an immature teratoma, it has this embryonic tissue in it, and it's nasty. The cause of these is due to meiotic failure. So with that, that's unlike the other cancers that we looked at. We have mostly been talking about cancers with mitotic failure. This one is incorrect meiosis. Compared to the mature teratomas, the immature have a, or I guess compared to other cancers, other malignancies, the immature teratoma has a low somatic mutation rate. And uh, so what's happening is if they have that low, mitotic or somatic mutation rate, what's happening is that those meiotic failures just lead to more and more meiotic failures, which end in a loss of heterozygosity, which we talked about earlier. So you have a loss of like the normal genes that are supposed to do the correct thing, and then you only have the bad ones. And that's something that we did see with the mitotic non-disjunctions, of course. Because meiosis is basically mitosis twice, there are two areas where a non-disjunction can occur. And when it comes to immature teratomas in the ovaries, a gene called BMP15 is often mutated in the germline, meaning that the mutation happened in the egg and sperm that made you. That's the germline mutation. It is inherited, and then the mutation, even though it's not present in your parents' whole entire body, it is present now in your whole entire body and your germline. This BMP15 is needed for egg development and maintenance, basically. So it makes sense that maybe it's potentially mutated in immature teratoma. The last important feature of immature teratoma is the presence of, already talked about that, the immature tissue. How it shouldn't be here. Oh, but I bring up another gene about the pluripotency. So again, neuroepithelial tissue should not be in adults. There's a gene called OCT4, which is a pluripotency marker. And it's involved with the differential development of mesoderm and endoderm tissues versus the neuroectoderm tissues. So it basically helps to say which path a cell is going to go down. And the fact that immature teratomas show this OCT4 expression is interesting because it's actually supposed to be downregulated in the neuroepithelial tissue. And the fact that it's just weird, the fact that it's there. So I guess it tells me that, like, OCT4 is inappropriately promoting pluripotency in these cells and preventing them from becoming the full neural tissues. Whereas if there were no OCT4, they would be allowed to not be pluripotent anymore. They would commit to their actual cell line. So interesting. The last and grossest teratoma that I'm going to talk about is the just straight up mature teratoma. specifically the dermoid cyst. I'll warn you when it's time for nasty pictures. It's not going to be on the next slide, but I will warn you. Dermoid cysts. They're mature teratomas that are generally benign. They're not usually malignant, and they have very ectodermal properties, as we shall see. Because they have hair and teeth and nails and shit. They're disgusting, and they truly live up to the name teratoma. They are congenital in that they do start growing before birth, like during development. Pretty much right here, like we were talking about earlier. That's what congenital means. It means that you are born with it. So people who have a mature teratoma are born with GCNIS. But it is not caused by a germline mutation. It is something that happens during development, a weird mutation or an aberration that starts during development. It's not a mutation in the egg or sperm that are contributing to the person who develops this. I'm mostly talking about ovarian dermoid cysts because they are well, they're the most interesting, if I'm being honest. And this GCNIS that eventually becomes an ovarian teratoma, it lies undetected until a mean age of 30 years old. That is generally when people with ovarian teratomas find out that they have an ovarian teratoma. Yeah. Yeah. That's the average. Isn't that fucked up? Oh man. How do these shitheads happen? Multiple ways, but mainly meiotic failure. Here's normal meiosis. I have not gone through meiosis, so I'll do it really quick. The oogonium, which is the reserve that you have at birth, undergoes some mitosis. Blah, 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 blah. And there's like, I don't know, a bunch of them. And after mitosis, they become the primary oocytes. And at this point, like I said, this occurs before birth, and the primary oocyte stays in meiosis 1, in prophase. It stays right here. It is only during leading up to ovulation, during puberty, and after puberty, obviously, where meiosis 1 will be completed, and we will get a secondary oocyte. This secondary oocyte has it's haploid, but it has duplications of each of the 23. It's not diploid. It's still N. It's one copy of each, but they are duplicated. It is literally not, like I said before, straight up fertilization is when meiosis is completed. They stay in metaphase 2 of meiosis until fertilization, when they actually become haploid. Only one of them. The other 3 in these divisions, when it comes to oogenesis, are polar bodies. They don't do anything. So however many kids your ovary parent has, you can pat them on the back and say, "Good job. You've fully completed meiosis twice," or whatever. So dermoid cysts happen because of this process being fucked up. In meiosis 1, if you have a failure in meiosis 1, these are homologous chromosomes. Like it's 2 duplicated chromosomes. Unlike in meiosis, where it's just the 1, it's 2. So in meiosis 1, these 2 are codependent as fuck, and they go to one of the oocytes. That means that the rest of these will have that missing chromosome. They will not have whatever chromosome is missing. And then both of these eggs here, I don't know why I didn't draw this. What is wrong with me? I'm going to draw right now. So these have nothing, blah, blah, blah. These 2 will each have an extra copy of the chromosome, which we do not want. We want one copy because it's haploid. We call this, these eggs are 2n-1, and these are 2n+1. Down syndrome tends to happen due to meiosis 1 errors because you have 2 chances here to have an extra chromosome. So a sperm with 1 copy of that same chromosome is going to fertilize it, and then when they become a zygote, now you have 3 copies of that 1 chromosome, and if that 1 chromosome happens to be chromosome 21, then that individual will have Down syndrome. Meiosis 1, when it usually happens. Okay, so that's Meiosis 1 nondisjunction. Of course you can have it in Meiosis 2, because why not? So in this one, these guys segregated normally, and then this one segregated normally, but this one did not. These 2 chromatids, it's the exact same as Mitosis nondisjunction. It's the exact same. They both went to this one. Any of these have the potential to be polar bodies. So most problems are from with like fertilization and then like chromosomal aberrations in the progeny. Those are generally meiosis 1 because at least here you have a 50% chance of a normal number of chromosomes. But anyway, now you have 2 normals, 1 2n+1 and 1 2n-1. The mature ovum can also oopsie doopsie duplicate its whole fucking genome for no reason. Like it just duplicates it. And we say that that's parthenogenic because it has a full set of chromosomes. It's 46XX, all maternal genes though. Absolutely no contribution of paternal genes to this 46XX. I don't know why it happens. It just does. There's also the potential of the PGCs to straight up just not do meiosis at all, and they just do mitosis. Whatever. Lastly, I couldn't find a freaking picture of this, but 2 ova. So that's, like these 2 or these 2. They confuse together. And make a fully, also, I think parthenogenic, just a full maternal genome in the cell. So these failures of cell division, along with ignorance to signals and pluripotency factors, results in the dermoid cyst. This beautiful table here is just showing like how it's possible for the dermoid cyst. It's possible to tell sort of maybe how a dermoid cyst came to be, like which of these mechanisms caused it, based on comparing the karyotype of the tumor cell and the karyotype of the person that the tumor was found in. It's not foolproof, as we'll see, but it is a fun little discovery. So meiosis 1 errors are the 2 different cells, the tumor and the regular. They tend to be heterozygous, and then down the line, they can be either. And by down the line, I mean physically. The centromere is at the middle of the chromosome. Those genes tend to not change. They tend to not recombine at all. So it's a pretty good indicator of like genes that those genes will probably stay put. So if they're heterozygous, they're going to be heterozygous in all their copies of the cell at the centromere. Things are different farther away from the center because those genes can recombine. So in meiosis 1 error, the teratoma tends to be different from the host. In meiosis 2, it's the middle of the chromosome are actually homozygous, which is pretty consistent with like how genes are at the centromere, and given that in meiosis 1 the chromosomes interacted in a more correct way. So right here they segregated correctly. So it makes sense that the genes are more consistent with the host. Whereas down the line, and again, this is recombination. The farther we get away, it's a little different. Now here's the really interesting thing with endoreduplication, which is when the ovum just duplicates its whole genome for no reason. That is all gonna be homozygous. Like they're all just gonna be the same allele. And then if the PGCs never did meiosis, then the genomes would actually be fully identical to the host. And then the fusion of two ova is weird. I don't understand this one. It's half and half heterozygous, like perfectly 50%. I don't know why that would be. But it's cool. I think this is really cool, but and it's important, but as you can see, it's still hard to differentiate between some of them because it's like, well, these are the same amount of heterozygous and these are the same amount of like homozygous. So yeah, types 1 and 4 are very heterozygous. But anyway, I just wanted to just list also just some fucked up chromosomal aberrations before I'm gonna show the pictures. I'm gonna show really gross pictures. But I just wanted to show gross chromosomes first. So this one, there's one extra chromosome. Their count is 47, but in actuality, they kind of swapped. So normal sex chromosomes, they're missing chromosome 15. They have an extra chromosome 21. And then they have something called a marker chromosome, which is just a random, I think in this case, it was a chromosome 15, just like a piece of it. Gross. This one had trisomy X. It had 3 X chromosomes. This one, normal sex chromosomes with an extra 7 and 12. And then this one, this beast right here, we have tetrasomy X. We have 4 X chromosomes, and it shows mosaicism, which is actually something that's pretty common in other types of cancers and other tumors, is the amount of chromosomes in each cell will be very different. So in this sample, 11 of the cells were 46XX. They were normal. 33 of them had the 4X chromosomes with the missing chromosome 13. And then 3 cells had, holy shit, 93 chromosomes, 4X chromosomes with an extra 13. So these cells really are so bad at being what they're supposed to be. And again, this is beginning as a GCNIS. So there's these inborn things that are predisposing these cells probably to dividing incorrectly. And then not only are they totally fucking up meiosis, but the dermoid cysts, they have they have all these like ectodermal cells as well. It's freaking crazy. So after everything I've talked about tonight, I hope that you can see just how much goes into making sure that our cells are dividing with our whole genomes intact, and with the correct epigenetic markers, depending on the cell. It it really floors me how fragile embryonic life can be, and this misexpression of genes that are supposed to tell our cells where to go, our germ cells where to go specifically, can lead to messed up neoplasms like this. We are going to round off tonight with some nasty photos of a dermoid cyst that was recently removed from a very good friend of mine. These pictures contain pictures of the cyst, as well as a uterus, ovaries, and other related areas. So, if real pictures of those are gonna gross you out, I would not look at the screen. Here we go. We'll start with the top left has the like the grossest picture. So, this is just an absolute aberration of human development that was growing inside this person before birth, meaning you had GCNIS that was undetectable, because I don't think that you can see that on like an ultrasound, but it was there since before birth and it turned into this. So uh it wasn't until weird symptoms really that this came about. But so these labels are lovely because would not be able to tell what any of this is. Clearly this is an ovary that has been to oblivion. This is the cyst is like the cyst is the ovary at this point. They are, like, one and the same. Um, it's hard to see, but there is a tooth in there. Um, sebum, meaning, grovary, um, and hair. So this is all of ectodermal origin, hence the name dermoid cyst. So sebum comes from sebaceous glands, which are in skin. So it's like secreting normal skin oils. Um, and then the hair, I mean, Grovery is your favorite Sesame Street character. Oh my god. Um, this is the uterus right here. So you can see the ovary just overpowered the fuck out of this uterus. It also looks like I can't tell in the picture, but it looks like is there hair on the uterus? Or is it not? If there is, it's probably not growing out of it. It's probably just from this. Um, but this is the cyst. It's an ovary that has become a cyst. So within this ovary, there was some up problem with meiosis and potentially still germ cell migration. It's like they got to the right place, but they just fucked up. And then here we can see the comparison with the other ovary. This is the other ovary. We have the uterus and the cyst ovary. I can't say for sure if this is like a normal size ovary, but it's a lot smaller than the cyst. And you can actually see these are the oviducts. They're being stretched as a result of to like not accommodate, but like are being stretched as a result of the size of this ovary. This is very nuts. Very cool, but very nuts. Um, just crazy. Um, another thing I wanted to point out is how this ovary right here with the oviduct and the fimbria, that looks like it kind of looks like a testy, a testes with the epididymis. Which I just wanted to point out because despite how different these structures are, they come from the exact same place, as I've been pretty much talking about this whole time. Those things, despite becoming different things eventually, they come from the exact same place. Um, and I know I didn't talk much about testicular teratomas. Um, it's just because ovarian GCTs are rare and therefore kind of more interesting to me. And interesting in a personal sense. um, but yeah, they really are. I mean, especially if you look at the bipotential gonad before it gets any signals to become anything, it's like, oh, they're just all right there. It's really cool. Um, it so it kind of surprises me that ovarian cysts are rare because sperm are actually way better at meiosis. There are less meiotic errors in sperm versus egg for some reason. So yeah, sperm are better at meiosis, but I think there's just still more chances of neoplasms to pop up in adult testes versus ovaries. That would be that'd be interesting if there was just this one quality that was missing. Like, we get teeth in ours, actually. It's way more fucked up. Um, I also think that they're rare in ovaries for some reason. This is just a spitball. This is pure spitball, but some seminomas are associated with a section of the X chromosome called XQ27. Um, and I think that because people with testes in general are hemizygous for the X chromosome, they have one copy, uh, that there could be an X-linked component where, they don't have an additional copy of the X chromosome that has a good copy of whatever the problem gene is. But I don't know. But yeah. Thanks to fucking Lugosi Rigatoni over there for these fucking disgusting pictures. This one is cool because it shows the hair and everything, but there's just something really charming about watching this poor oviduct like like really try to like hold on to this this fucking shit right here. It really cracks me up. Yeah, like, huh, what's going on here? And it's so interesting the combination of um like you have this you clearly you had to have had this GCNIS in your body beforehand, but the instead of the chromosome 12p that launches into like the germanomas and whatever, it seems like it's just another like meiotic failure because you're not doing meiosis yet, at week 4 when the GCNIS um is occurring. So it's like there's clearly something different that goes on in teratomas that pushes it into existence. And because they don't have the isochromosome 12p, I'm thinking that it's just there's another disjunction, meiotic disjunction that just happens, and it likely won't happen until show up until after puberty. Like I think the reason why people in their 30s it happens to them is because you've had enough menstrual cycles and you've had enough like hormones go through there that some eggs are being chosen, some are being brought out of meiosis 1, and this is when they're actually like undergoing multiple divisions or whatever. So I think that makes sense. As fucked up as it is. Ugh. I don't have a final slide, I don't think, so I'm just gonna have this up. No, I'll do we'll have a whole meiosis up. I just don't want to have the gross stuff up for people, but thanks everyone for hanging. Thank you to my buddy for the the dermoid cyst. Thank you for the dermoid cyst. You give it to me. I I love it. No, your doctor is the freak. Your doctor weren't they like, oh my god. Did they do karyotyping on it? Did they like do any testing on it? Because that would be really interesting. What if you're this guy? What if you're fucking 93 chromosome mosaic fucking teratoma? If there are questions, ask them now because I'm about to pop out. You know what's funny is there's fetiform teratoma and then there's fetu in fetu, which is like there's like a heavy debate on like what the differences are between those two. I imagine fetu and fetu is like that's more of like a parasitic twin kind of situation, but yeah. 2 ovaries and 2 adrenals in that freaking homunculoid. So gross and weird.